Pure cultures of bacteria are most often obtained by the streak plate technique. In this procedure, an inoculum, a mixture of two bacteria in nutrient broth, is placed on a solid nutrient medium and then repeatedly diluted to separate individual cells. You will inoculate the agar using a 4-quadrant streak as shown. Use a fine-tipped Sharpie to divide the bottom of a nutrient agar plate into 4 quadrants. Label the bottom of the plate with your initials, the date, the type of medium, and the culture or source of the inoculum. You don't want to label the top of the plate because tops can get separated. Use a sterile loop to obtain the inoculum. We will use a loopful of Staphylococcus aureus. Flame your loop until it is red hot and let it cool. Remove the cap from the tube, pass the tube through the flame, obtain a loop of bacteria from the tube, and then pass the tube back through the flame before recapping. Lift one side of the Petri plate lid and streak about 1/4 of the agar with as many back and forth streaks as possible. Hold the loop like a pencil and gently touch the surface of the agar. Replace the lid and flame the loop. In the next steps, we will spread the bacteria from the initial inoculum into subsequent areas of the agar plate. Always let the loop cool before performing the streak. Using the sterilized loop, streak through one area of the first sector and then streak a few times away from the first sector. As shown in the illustration, there is only one streak through the initial inoculum. Replace the lid and flame the loop. Using your sterilized loop, streak through one area of the second sector and then streak a few times away from the second sector, as shown in the illustration. Replace the lid and flame the loop. Using your sterilized loop, streak through one area of the third sector and then streak a few times away from the third sector, as shown in the illustration. Replace the lid and flame the loop. Incubate the plate in an inverted position at 35 to 37 degrees Celsius for at least 24 hours. During incubation, water evaporates from the agar and will condense when it reaches the cooler Petri plate lid. If the plate is inverted, this condensation will stay in the lid, away from the agar. If the plate is incubated with the lid on top, the condensation will rain back onto the agar. Bacteria will move in the water, and you won't see discrete colonies. Individual or small groups of cells will divide, producing a visible colony-forming unit, or colony. Observe areas of well-isolated colonies. Different species will produce colonies that appear differently. This plate has a pure culture. All the colonies are identical. There are 2 different bacteria on this plate. One produces red colonies, and the other makes smaller, yellow colonies. After recording all of your data, properly discard your plates to be autoclaved.